Final speaker today is Dr. Timothy Yu. He is a neurologist and researcher in the Division of Genetics and Genomics at Boston Children's Hospital. He completed his MD and PhD at UCSF, neurology residency at Massachusetts General Hospital and Brigham and Women's Hospital. His group specializes in genetics and neurobiology of neurodevelopmental disorders and genomic medicine for rare diseases. Please welcome Dr. Yu.
Hi, thank you very much. That's fine. That's fine. I'd like to start by thanking the Science Committee for the very kind invitation to come share our work with you. This is my disclosure side. I have nothing to disclose. For context, I'd like to explain a little bit. My lab at Boston Children's has, for the last 5 years, been working on using genomic tools to explore the basis of human brain development through the lens of rare disease. Today, I'd like to share with you an interesting patient that I've had the privilege of working with that is extending our interests from diagnosis towards questions of therapy. And to set up this question, I think of this as a very interesting time in genomics when the tools at our disposal are making it increasingly easy to come to diagnoses for very difficult neurologic diagnostic dilemmas. However, the problem still remains how we turn these diagnoses into treatments. Now, I'd like to introduce a family to you that it's possible I realize some of you may have heard about. There's been a little bit of press. But for those of you who don't know her, I'll introduce her to you with the family's permission for the first time. This is my patient, Mila. She is an 8-year-old girl, and she hailed from Colorado. And as a young child, she had a very typical energetic, vivacious, and bold development. But beginning at age 4, she began developing some worrying symptoms. This began with low light vision problems at age 4, some stumbling and tripping beginning at age 5, and then at age 6, very worrisome accelerating symptoms of neurologic regression, worsening gait, speech, behavior regression, and her vision precipitously failed that summer. The ensuing hospital admission led to more concerning findings: retinal degeneration, cerebellar atrophy, subclinical seizures, and eventually led the clinical team to a diagnosis of Batten disease. Now, as you all know, Batten disease is a family of disorders, recessive disorders, caused by mutations in one of 14 different key lysosomal genes. And her particular subtype was CLN7. This is a particularly rare type of Batten disease with only about 70 patients described in the literature. And the age of onset matched her symptoms around age 3.5, and it invariably progresses with vision loss, seizures, motor, and cognitive decline, with a typical age of death of around 11. So, strictly speaking, of course, this was a terrible, terrible diagnosis. Now, there were no treatments for Batten disease, but if you've read a little bit of the press, you know how remarkable our family was. Within 2 weeks of arriving at their diagnosis, the family launched a foundation to try and raise awareness of Batten disease, to try and share their story with others, and to try and raise money to launch a gene therapy for an AAV-based CLN7 gene therapy project. Now, however, I'm not speaking to you about gene therapy today, and the reason is because in this case there was a wrinkle. That wrinkle was that she had only arrived at half of a diagnosis. Her geneticist, Dr. Larson, had sent clinical genetic testing for the CLN7 gene locus and found only a single mutation, a known pathogenic one, but only one. And I told you this is a recessive disease. So where's the second hit? Dr. Larson counseled the family that to solve this completely, that they would need to have whole genome sequencing. Now, this was 2016. The question was, where could they do that and how quickly could that happen? And that's where my lab came in. It turned out that on a Friday night, January 2017, a month after her clinical diagnosis, my wife turned to me and shared with me this Facebook post. It described a post by the mother's college friend who had posted in a group called PMG Massachusetts. This is a medical advice and referral sharing group on Facebook called the Physician Moms Group. And it described her college friend's situation, their daughter's recent diagnosis, and the fact that they needed whole genome sequencing, but they needed someone who could expedite that for them. And our group had done a lot of whole genome sequencing, and after reading about the case and hearing about what they were trying to do, we decided that we wanted to reach out and try to help. So we reached out and offered to do whole genome sequencing for our patient and the entire family. And a month later, we found something rather unexpected. Instead of finding a point mutation in the CLN7 gene to complete the diagnosis, we found actually a retrotransposon insertion. Now, what's that? What's a retrotransposon to? Remind you who may be a little bit rusty on retrotransposons. These are jumping genes. These are unusual jumping genes that live in our genomes. Like other genes, they can be transcribed into RNA, but they have the unusual property of being able to be converted back to DNA and then copied and pasted into new sites. This doesn't sound like a very good idea for the genome, but luckily most retrotransposons in the genome are actually quiet, extinct, sort of like ancient volcanoes. However, occasionally some subfamilies of retrotransposons can become hot and active, and they can pick themselves up and land in new places. And if you're unlucky enough that it lands in a critical neurogenetic disease gene, it can break that gene and cause a disease. And so we had discovered that this is exactly what had happened to our patient, that this was a deep intronic insertion of a retrotransposon, explaining why it was missed by clinical sequencing, and it actually had the impact of activating a cryptic splice site. It created a new splice site, deep in intron 6, and that created, in turn, a pseudoexon, a poison exon, because the very second codon of that new exon was a stop codon. And we confirmed this by RT-PCR and RNA-seq and splice junction analysis. So this was our second hit. 2 mutations, recessive disease, case solved. Well, what next? Well, this was January, February, March 2017, and we asked the question, could we actually fix this? The principle was, could we use an antisense oligonucleotide to block the abnormal splicing and restore the normal gene pattern of splicing in her cells? And would that be of some therapeutic benefit? Now, we were only emboldened to even think about proposing this because, of course, December 2016 was when Spinraza, or nusinersen, was approved for SMA. And we were all reading about the wonderful successes of this antisense oligonucleotide drug for an autosomal recessive neurodegenerative condition. And this is a drug that's delivered by lumbar puncture and has had remarkable clinical effects in the patients who've received it. And the proposition was, could we offer something similar to our patient? The parallels between the genetics, the pathophysiology were clear. We could look at the chemistry of the nusinersen drug and ask whether we could design a version of that using the same chemistry but only matched to our patient. And we would deliver it intrathecally, and hopefully it would activate the inactivated CLN7 gene locus. Of course, we would have the benefit of looking at the literature to help us along the way and calling up good colleagues who helped us with initial advice. And the plan that we devised was as follows. We decided to try to find N of 1 customized therapies by designing a patient-specific screen using patient fibroblasts, bioinformatic prediction of splice sites, synthesis of probe oligonucleotides, introductions of those probes into the cells, and then assays to see whether or not we were improving cellular function. We quickly landed on some hits. This is an RNA analysis which showed on the top left here patient fibroblasts without any treatment. And you see that there's a top band and a lower band, and the lower band is the abnormal band. And you see that that abnormal band predominates. It's present in a 1-to-2 ratio relative to normal. But once we introduced one of a series of exon-skipping antisense oligos, one of it was flipping that ratio, giving us 3 times more normal than abnormal. We repeated this in multiple replicates and different types of cell lines. We did RNA-seq analyses to convince ourselves that this was topologically accurate. This is an experiment that I won't go into the details of because there are a lot of numbers, but essentially between 7 and 11% of the transcript was correctly spliced in her, and administration of our oligonucleotide boosted that by about 3.5-fold. And this was about the range of effect that Spinraza had shown in the early papers in the 90s, demonstrating its impact on the SMN2 locus. So we thought comparatively we were doing pretty well. Well, that's RNA. Well, what about protein function or cellular function? Well, we found supportive data from patient fibroblasts looking at lysosomal electron microscopy. Here is a normal fibroblast showing the normal topology under EM. However, in our patient cells with the CLN7 mutation, there's an accumulation of these white droplets which are built of storage material, abnormal storage material in her lysosomes. And treatment of her cells with our oligonucleotide greatly reduced the number and the size of those storage droplets. Our critical collaborator, Joe Mazzulli, also applied a number of other biochemical assays that gave us the same consistent response. With every measure he looked at, our oligonucleotide was improving lysosomal function and cellular partitioning. December 2016, this is when our patient was clinically diagnosed. January, we became introduced to the family. February, we finished whole genome sequencing. March, we found the retrotransposon. In April, we were busy generating patient cell lines. In May, we found proof that it was altering splicing. And in June, we began to say, hey, maybe we have an idea here we should talk about. And we began discussing this carefully with our family and her clinician in Colorado. In July, we were working on designing and ordering oligos. We began August to talk about this with the FDA. In September and October, we found evidence that it was working at the RNA and the cell level. We're feeling pretty good, but there's much more to be done. We had only gotten through proof of concept. How are we going to navigate the rest of the steps of a traditional drug development program? Well, we began calling around, and we began checking in with our family to motivate folks to come help our cause. During this whole time when we were working hard, our patient continued, unfortunately, to decline. And the parents of our patient wrote this letter to try and bring to the FDA's attention how critical her need was, with loss of function in many different domains: vision, speech, walking, strength, and so forth. And so we set up regulatory collaborations with CROs, CMOs, and the FDA to find folks who could help us navigate this first path. And we used an individual patient expanded access investigational new drug application as the carrier for our package. And with lots of help, we assembled an aggressive timeline to bring our drug from concept to delivery to our patient, in theory, in as little as 4 months, with manufacturing, release testing, a modicum of animal safety testing, and an overlapping clinical trial with appropriate FDA approval. Manufacturing was completed on December 12th, 2017. Animal toxicity studies were launched a week later in Montreal with intrathecal injections in 3 cohorts of rats at multiple doses. And this data was examined and put together and given to the FDA, so they had an early heads up as to how our drug was doing. We developed a clinical protocol at Boston Children's. I, with my colleagues listed here, designed a gradual dose escalation protocol with administration every 2 weeks, maintenance dosing after a careful ramp up, and mimicking our protocol after the Spinraza nusinersen clinical trial designs, with appropriate safety monitoring and some early exploratory outcome measures. After a review of the animal safety data and our clinical protocol, the FDA granted us officially permission to proceed on January 19th. We actually hadn't received the drug yet. That came about a week later. It finished clearance from the final release tests. And we launched our trial on January 31st with giving our first careful doses to our patient. Now, how are we doing? We are a little over a year into our trial, and I've shown here the list of doses that she's received with the gradual dose escalations. I'll note that by week 3, by dose 3, we had slightly exceeded the typical Spinraza nusinersen dose, and we continued to climb up, ultimately up to 3 times higher than that. And we did a series of evaluations to monitor both for safety and for efficacy. I'm happy to say that at the 1-year point, safety and adverse effects, the safety has been well tolerated. There have been no adverse effects attributable to the drug to date. Outcomes, this is a single patient. It's difficult to stand up here without statistics and tell you exactly what's happening, but I can tell you qualitatively that the family has reported substantial stabilization along many domains compared to the steep decline that she was exhibiting the months leading into our trial. What objective data do I have to support that? We have Vineland scores and GMFM motor function scores, which show that same pattern, declines in the period beforehand, relative stabilization in the year since. We've also carefully been following seizure logs and video EEGs. This is a graph of her seizure frequency and intensity over the first 3 months. And what we're seeing here plotted are this number of seizures per day on the axis on the left. The escalating doses are in the red on the right. And the color coding of the circles indicates how long the seizures are lasting. You can see at the beginning there were up to 30 per day, with almost all of them lasting greater than a minute. And what we've seen over the course of the dose escalation is the frequency has gone down and the intensity has lessened so that almost none of them now are greater than a minute. They're only lasting just a few seconds. As we space the doses out in the subsequent year, you see the data gets a little bit noisier, and the frequency bounces up and down a little bit. And so we are thinking about adjusting our intervals of dosing. But the intensity remains lower than before. So we think of this as very promising early signs, and we will be following for quite some time. I'd like to close with a few conclusions. This is an N of 1 drug. This is a mutation that we identified that we have not seen in any other patient with Batten disease. We've not seen it in the general population either. And so this drug is only applicable to our patient, not others with Batten disease, not others with CLN7 disease. But we do think it has us thinking hard about potentially broad implications. If it's possible to turn around drug development in such a short time when things line up, could we work towards a future when we have rapid genetic therapy clinics that are designing customized drugs for patients in dire straits? That idea is based off of the simple fact that nusinersen has the sequence shown here. Melicin, our patient's drug, has the sequence shown here. And that they share many properties based on their backbone and their chemistry, but less dependent on their sequence. So we have an idea for putting together a pilot to develop personalized therapies for other patients who may be in similar situations. We are having early discussions with the FDA about this idea, with ethicists, with patient foundations and colleagues. And this is work that we're very excited about, and I hope that in the months and years to come, we'll be able to provide you with more updates as we go. I'm going to pause. I'm a little bit over. I apologize. But these are my acknowledgments. It takes a village to pull together work like this so quickly. And there's a huge host of folks from academia, within the hospital, between hospitals, from industry, and various contract manufacturers and folks who volunteered their time to make this first patient happen. And we hope that it might open doors for others in similar situations. Thank you.
Thank you. A popular question from the audience is, are there other cases of retroposon pathogenic variants causing NCL or other neurodegenerative disorders?
Right, that's a really good question. So one of the lessons of this case is that you don't know what you're missing until you go looking for it. And there have been published in the academic literature several dozen cases of mobile DNA elements, retrotransposons, causing human disease. None, to my knowledge, are specifically for Batten disease. And for our particular patient's retrotransposon, this SVA type of retrotransposon, there are at least 13 other cases published in the literature. But the clinical diagnostic labs are on to this, and they are beginning to develop algorithms for picking these up, and those will be important to help complete, oftentimes, that 10% of patients that you know clinically what the diagnosis should be, but you just can't seem to get the full molecular picture.
Thank you. Thank you for joining us. This concludes the 2019 Hot Topics plenary session. We will now take a short break before we welcome Hugh Herr. Dr. Herr's special presentation on the new era of extreme bionics begins at 5:45 p.m., followed by the annual meeting opening reception.